Hello students, today in this biochemistry class we will discuss about the enzymes. In this introductory portion, first we discuss about the biomedical importance of enzymes, basic concepts about enzymes, classification of the enzymes as per IUB, and here IUB stands for International Union of Biochemistry. At first we have to know what is the enzymes. Now enzymes are the biologic polymers that catalyze the chemical reactions that make the life as we know it possible. The living cell is the site of tremendous biochemical activity called the metabolism. This is the process of chemical activity called the metabolism, and by which physical change which goes on continuously in the living organism, building up of new tissue, replacement of the old tissue, conversion of food to energy, disposal of waste materials, reproduction, and all these activities that we characterize as life are possible with the help of the enzymes. In their chemical nature, we all know that the enzymes are the protein and they are high molecular weight compounds. Enzymes can be denatured and precipitated with salts and solvents and other reagents, and for their activity there should be certain conditions of temperature and the pH. Many enzymes may require the presence of the other compounds. They may be termed as the cofactor or the coenzymes. Here we can see that two figures. One is termed as the cofactor or the coenzymes, and another is termed as the apoenzymes. Now generally we said the enzymes, but in scientific or the biochemically it can be said the hollow enzyme, means without a cofactor a portion of the enzyme is termed as the apoenzymes. When the cofactor attached with that and ready for the binding of substrate, it can be termed as the hollow enzyme. Now we'll discuss about the biomedical importance of the enzymes. Enzymes catalyzes the chemical reactions, participate in the breakdown of the nutrients, and help to supply energy and the chemical building blocks. Enzymes are responsible to assemble the building blocks in the proteins, DNA, and membrane cells and tissue. Means enzymes are help in the synthesis of these biomolecules, diagnosis and the prediction of the many diseases can be done by the presence of the enzymes. Now these are the earliest studies about enzymes. In 1835, a Swedish chemist, John Jacob, who termed a substance as the chemical action catalytic substance, and this is the first indication of the enzyme. But in 1926, at first time, James B. Sumner of Cornell University discovered the enzyme, and he is able to isolate and crystallize the enzyme urease from the jack bean. Here we can see the picture of scientist James B. Sumner and a picture of the jack bean. Now we will discuss about the enzymes and its specificity. Enzymes shows their specificity among the substances, and they are highly or extremely selective for their substances. Enzymes are specific not simply for the type of the reaction but also for a single substrate or the set of closely related substrates. Enzymes are stereospecific also, and in general There are four distinct types of the specificity. Here it can be grouped as the absolute specificity, group specificity, linkage specificity, and the stereochemical specificity. Now we discuss about the absolute specificity. Here the enzyme will catalyze only one type of reaction. This can be termed as the absolute specificity. Whenever we discuss about the group specificity, the enzyme will act only on molecules, only a single molecules that have the specific functional groups such as the amino, phosphate or the methyl groups. This is termed as the group specificity. Whenever we discuss about the linkage specificity, the enzyme will act on a particular type of the chemical bond regardless of the rest of the molecular structure. Means a specific enzyme act on a specific bond, but where the stereochemical specificity is discussed, the enzyme will act on a particular optical isomer. Means a stereoisomer. Enzyme act on a specific stereoisomer that can be termed as the stereochemical specificity of the enzymes. Now the naming and the classification as per the IUB, International Union of Biochemistry. International Union of Biochemistry developed a system of enzyme nomenclature in which each enzyme has a unique name and code number that identify the type of the reaction catalyzed and the substrate involved. As per this system, the name of every enzyme starts with the capital E and C and followed by the four digits. In among them, first digit represent the class of the enzyme, second digit represent the subclass of the enzyme, third digit represent the subclass or the subgroup of the enzyme, and the fourth digit give the number of the particular enzyme in the list. And this is the classification as per the International Union of Biochemistry. Here we can be able to group enzyme into 6 classes. First one is the oxidoreductase, second one is the transferase, third one is the hydrolase, fourth ligase, fifth isomerase, and sixth is ligase. Now oxidoreductase, they are responsible for catalyzing the oxidation and reduction reactions. Now the second one is the transferase. They catalyze transfer of the moieties such as the glycosyl, methyl or the phosphoryl groups. Third category is the hydrolase catalyze the hydrolytic cleavage of the carbon-carbon, carbon-oxygen and carbon-nitrogen and other bonds. Lyase catalyze the cleavage of the carbon-carbon, carbon-oxygen and carbon-nitrogen and other bonds by atom elimination and leaving double bonds. Isomerase catalyze the geometric or the structural changes within a molecule. Ligase catalyze the rejoining together of two molecules coupled to the hydrolysis of ATP. Now this is the naming of the hexokinase, very popular enzyme, and this is the IUB's naming of the hexokinase. The simple name is hexokinase, but as per IUB it is the D-hexo-6 phosphotransferase EC2711. This name identifies the hexokinase as a member of class 2, means transferase. Second digit 7 indicates the subclass, that is the phosphoryl group, and the third digit 1 represent the subclass. Here alcohol is the phosphoryl acceptor, and the hexo-6, this term indicates that the alcohol phosphorylated is on the 6th carbon of the hexose. At last we can see the structure and the action of the hexokinase. At the right hand side we can see the 3D structure of the hexokinase enzyme, and this is the pathway by hexokinase act on the hexo sugars and produces the hexo 6 phosphate by accepting a ATP molecule, and after that from that hexo 6 phosphate glycogen, starch, and the glycolysis and pentose phosphate pathway begins. Thank you.